But this next talk is really meant to explain what is UBE3A. We all say it, we've all learned to say it, but do we all understand actually what it does and why it's so important and why ensuring we have UBE3A can help to improve the lives of those living with Angelman syndrome. So I'm excited for you to hear Dr. Seth Margolis from Johns Hopkins University. I was lucky enough to hear him speak at FAST Italy last year. And when I heard his presentation, I was incredibly touched by the intricacies of his knowledge of UBE3A. And I've been working now on UBE3A for almost a decade, and I haven't heard someone so elegantly explain what it does. So I thought it was really important that I didn't only hear that, but you all heard that. So I've tasked him with explaining it in a way that we all can understand, because it's complicated, it's super complicated. But I think he's gonna do that. I previewed his slides and they're excellent. So I have no doubt that you're gonna accomplish this. And you can also explain that your wife is a school teacher, so you promised me she was going to give a thumbs up to your slides before you actually gave them to our community. So Seth, thank you.
All right, thank you. Yeah, thank you. All right. Okay. All right. Thank you to Allison and to the organizers for the invitation. And thank you, Allison, for that nice introduction. As Allison said, today I'm going to talk to you about UBE3A. And I think before I get into that, I wanted to give some context. So our nervous systems, central and peripheral nervous systems, are made up of neurons we're all very familiar with. And they're really critical for our behaviors, controlling our behaviors and sensing our world. And within our neurons, in all of our neurons, and many of you have already now seen from several of the slides in previous talks, these beautifully colored images, these are proteins. Within all of our neurons, these are proteins. And scientists have the capacity to color these proteins. So, and I just name a few up there, and the specifics aren't really important. What's really important here is just to realize that within our neurons are these proteins, and these proteins are what's doing the functions of our neurons. So the first sort of take home is neurons contain proteins that do the work. So proteins aren't just there. They don't just pop up from nowhere. Proteins are made. Proteins are made in all of our cells, okay? And I'm not going to go through the details of these complex molecular events that make our proteins. But on the top part here, where I write nascent protein, that's the birth of a protein. And then there's the process where it's folded, and eventually in the folded protein, that's the protein that goes on and does all the important functions of all of our cells, including neurons. And throughout this process, our cells have this amazing capacity to determine whether the protein's being made properly, or at a specific time, whether we no longer need that protein. And that's this process where you see the red being broken up into tiny little lines. That's protein degradation. And this process in whole is referred to as protein homeostasis. All of our cells, neurons included, want to make sure that our protein levels, both the proteins that are made and the proteins that are degraded, are kept at an even level. It's very well regulated. So the next take home is proteins are made and removed in our neurons. And this removal process is controlled by this incredible structure within all of our cells, including neurons. This is referred to as the proteasome. It's something my lab studies in great detail. And it might look complicated, and I'm not going to get into all of the details of what you're looking at here, but I think you can appreciate that this picture of this 20S proteasome on the left looks kind of like a rectangular barrel. And if we flip it and look down the top, there's a hole there. And that hole is where our proteins go to get degraded. So it's, in some regard, referred to as the trash can of the cell. So I'm going to go back. Okay, so the protein complex here that's degrading our proteins is really important for cell function. including neuron function. And all it does is degrade proteins. So it needs help in determining which protein should we degrade. Otherwise, it would just degrade every protein that it came near. So these larger structures, these gray structures on the top, these are referred to as caps. It looks like a cap. It sits on the top and the bottom. And these structures are very important for unfolding our proteins. Remember you saw that red folded protein? Proteins are in that folded shape. These structures unfold the protein and put it into that pore so it can get degraded. So proteasomes remove our proteins. Those are the key players. These are the key critical players in this protein homeostasis process. And they're really important for nervous system function. So here's a neuron. It's flashing, it's active, it's functioning. Okay? If I inhibit the proteasome using a small chemical inhibitor, you get a very small change in activity very rapidly. And this is to illustrate, here quantified, that a proteasome functions to control the neuron, and if we inhibit it, it very rapidly can change that neuron's function. Okay, so simply, no proteasomes, you get no protein removal, and the neurons don't function well. So the proteasome's ability to control protein degradation is critical. And as I just mentioned, it doesn't just function all by itself. It needs to be told what to degrade. We don't just degrade every protein, just as you wouldn't just throw everything in your house into the trash can. It needs to be told what to degrade. And that's largely controlled by this process. This is the ubiquitin proteasome system. And many of you may already be familiar with this. And there's a series of steps that I just want to quickly take you through. So there is a substrate. A substrate is the protein we want to degrade. And then there are these other molecules, E1, E2, and E3. They work together to associate with that substrate and put that protein into the proteasome to degrade it. And the E3, that's what UBE3A is. UBE3A is the E3 ligase. It's one of 600 E3 ligases in our genome that helps to target specific proteins to go to the proteasome to get degraded. And so what I want you to appreciate is that UBE3A in our cells, in our neurons, tags some proteins, not all proteins, but specific proteins to be removed by the proteasome, and that's good for the neuron. That's the key appreciation for understanding UBE3A. And there's a lot of mechanistic detail about how it does that. We're not going to get into that here, but I'm going to tell you more at a high level about what are the types of proteins that UBE3A might target, how do we go about finding that, and how may that give us insight on how, when UBE3A is gone, you get such a broad change in developmental function. So a couple other details that I want to point out. Neuron is a complex cell type. You've seen pictures of these before. You have a soma, a nucleus, this sort of triangular shape at the bottom, but then you have these long processes. And there are proteins across the entire neuron, including inside the nucleus. And proteasomes are located everywhere, and so is UBE3A. And you can have ubiquitin-mediated proteasome-dependent degradation at all of these locations. And what's not always so obvious when you look at these images is that the substrates, while the proteasomes and the E3 ligases are everywhere, the substrates in these different locations, remember those are the things that want to be degraded, are different throughout the cell. I also wanted to make this point, which is UBE3A is an E3 ligase. It's disrupted in Angelman syndrome. And there are other E3 ligases that have also been found, when mutated, lead to other neurodevelopmental disorders. And so a big question in the field, and something my lab is particularly interested in, is to understand what protein substrates of E3 ligases are relevant to human health. And in particular, Angelman syndrome. What are the substrates of UBE3A that give rise to phenotypes that lead to neurodevelopmental dysfunction? And I'm going to tell you towards the end of why I think that has some value in thinking of leading to potential treatments. So I'm going to sort of skip over a lot of years of data, and I'm going to share with you at least one substrate first, and then I'm going to tell you how we went to find every substrate we could possibly find. So as a postdoc, when I was working with Michael Greenberg at Harvard, we had discovered this protein referred to as affexin 5. And this protein played a very unique function in the nervous system. So these neurons are, what we're looking at here are fluorescent neurons. These are expressing this GFP protein. And we could quantify the number of contacts these neurons were making with other neurons. And what I hope you can appreciate just from this bar graph is that if you remove affexin 5, you get way more contacts. And it seems like it's a good thing, but it's actually not the ideal situation. So when we looked at this protein, affexin 5, labeled E5 on the top, I hope you can appreciate, and this is a developmental representation of looking at affexin 5 at different stages in development from neurons, and you can see that protein band at the very top going away. That's this protein getting degraded. So naturally, this protein gets degraded, and that's something we learned as we were doing research on this protein. So affexin 5 blocks a neuron, and we found was degraded by the proteasome. And it turns out, with further research, that UBE3A was the E3 ligase targeting this protein. And what I'm showing you here is that when I mix UBE3A with affexin 5, you get this smear, this band that sort of rides up on the gel. And that's an indication that this thing is being ubiquitinated. This is being targeted by affexin 5. And in animal models where we remove UBE3A, if you look in the middle lane of the 3 lanes there, you can see there's more affexin 5. Okay, so this predicted that UBE3A targets affexin 5, that's what the data were demonstrating, to the proteasome, which we all learned about now. And so no UBE3A is too much affexin 5, okay? Which turns out to not be great for the neuron. And the reason it's not great is because when you have too much affexin 5, you actually have reduced synaptic contact. And so we predicted from a substrate sort of hypothesis that if we reduced the affexin 5 in the UBE3A animal models, we would potentially rescue behavior. And that's sort of what we saw. So these behavioral tests sort of evaluates the sort of cognitive functioning of the animal. And we learned on the left side here that the animals have reduced behavior compared to controls. That's these lower error bars. But that if we remove affexin 5, we could rescue that. And so reducing affexin 5 restores function when UBE3A is gone. And in a more general sense, it indicated the possibility that substrates might be, we know, are downstream of UBE3A, that when UBE3A is gone, those substrates are now deregulated, they're not being degraded, or they're not being regulated properly, and might be the foundation for some aspects of phenotypes observed in Angelman syndrome. So my lab became very interested in, like, what are all the substrates of UBE3A? A lot of technology today affords us the opportunity to sort of go after everything at once. So what this is sort of, this image here is sort of showing you is a sort of experimental outline of what we did. And I can take you through it briefly. So we have basically glass slides with 20,000 human proteins printed on the slides. We can then incubate, remember the E1, E2, and in this case UBE3A, and ubiquitin on these slides, and we could look at every protein that could potentially be ubiquitinated by UBE3A. And on the right is a heat map that sort of demonstrates proteins that are more ubiquitinated with UBE3A versus controls. So what do we learn? UBE3A has many possible protein targets. And it's hard actually to look at the data and easily separate out which are the most important targets, which are the ones that might have the highest relevance. And so you have to take your time to systematically think about this and use artificial intelligence or other types of approaches to really begin to understand what does all of this mean. And I also want to keep something in mind. Even though we see all of these proteins that can be ubiquitinated by UBE3A, it doesn't mean that they all are being ubiquitinated in our neurons equally well, or at least in the capacity that mediates effects on our biology. But it gives us a foundation. So when we look at all these different proteins, and we try to understand what are the different classes of proteins we're seeing, a couple things come out that are interesting. Many of them are expressed in the brain. Some of those substrates are associated with other neurodevelopmental dysfunction. And there are various pathways that might indicate a relationship between the loss of UBE3A and changes in some of the behaviors that we've seen, including motor deficits and others that I think this group will hear more about in the talks to coming and are very familiar with. So the sort of take home here is some UBE3A targets are known, sorry, that's a typo there, to impact various brain disease and be a part of many brain processes. Okay, so what do we do with this information? Clearly, adding back UBE3A makes the most sense. I think it's really the best way to go because it's going to take care of the loss of capacity of targeting these different substrates. But if we were to actually want to go after specific substrates, such as, say, Effexin 5, how do we do that? Is that even possible? That seems really hard. Well, I'll tell you that technology today is giving us the opportunity to do that. And some of that stems from the recognition and the realization, and AlphaFold was already introduced, that we know the structures or can generate the structures of every one of these substrates in association with UBE3A. So this is just an illustration where in blue is the UBE3A structure, and in orange is different substrates that we identified. Now, why is this important? Well, it's important because it tells us that UBE3A binds its substrates, as do all ligases, okay? And that means we can control that. We can control it by using or identifying small molecules that can bring together any two proteins together. And these molecules are referred to as molecular glues. So if our goal was to say we wanted to, in the absence of UBE3A, target one of the substrates, I could build a molecular glue that could bring a ligase that is available, that is still expressed, and target it to a substrate that UBE3A would normally target, but now can no longer because it's not there. And I just wanted to introduce that technology because I think it's relevant for this audience. So this comes from a collaboration with a team of Hopkins scientists that I'm introducing here, where we've spent the last 4 or 5 years, maybe longer, trying to build some technology at Johns Hopkins to find these molecular glue type molecules that would allow us to bring together any E3 ligase with any substrate of interest. And we do this in such a way where we can look at as many as possible. Okay, so drugging the human proteome. So I've already introduced this process to you to some extent, where you have a substrate, okay, and you have an E2, and then you're bringing that substrate all the way down to the point where you're getting to the proteasome, where you can then get rid of that substrate. And this little, I guess it's a yellow drug, that's our molecular glue. And our goal has been to identify these molecular glues that bring together an E3 ligase to a substrate of interest to drive it to the proteasome. So simply the way we do this is we can express a large number of different proteins. We can make any one of the 20,000 proteins in the human proteome. There's actually more than that because of isoforms, but we can make almost any of them and any E3 ligases. So in the context of Angelman syndrome, we've been making ligases that we know are available and still expressed, and we've been making substrates that we believe to be relevant to the biology. We can then do large-scale screening, and we can ultimately identify these small compounds that are effectively molecular glues that allow for degradation. And this is early days, and this is a complex process. But we've had some success, and it gives us some opportunity to think more deeply about the fact that there are druggable capacities that allow us to control normal cellular degradation, to remove target proteins with a purpose. And that would be an ultimate goal. And being able to test that purpose is of great value moving forward. So I want to summarize, getting us back on time here. I want to summarize. So neurons contain proteins that work. I think that's very important to recognize. The proteins do the work in our neurons. And proteins are both made and they're removed. And that's also really important to think about. And proteasomes do this process of removal. But you don't just remove any protein, right? You have to selectively choose that protein, okay? And another point I sort of skipped here, which is if you don't have the ability to remove the protein, it's not really ideal for the neuron. And the way to specifically remove a protein in the context of UBE3A is to tag that protein with ubiquitin, which then sends it to the proteasome, and that's what the neuron wants to have happen. Now, we've learned now that UBE3A has many, many targets, okay? And those targets, to some extent, give us insight into how the loss of UBE3A might give rise to a variety of different pathways. And reducing a UBE3A substrate, at least in the one example I shared, and there are others in the field, restores neuron function when UBE3A is gone. And that there's now new technology that even though UBE3A may not be there, that would allow us to still target those substrates by co-opting another ligase and using a molecular glue to send that substrate of interest to the proteasome. And with that, I will end a little early, but none of this work is done without the tremendous help from the researchers that come through the lab, as well as the funding sources. So, thank you.
That was an excellent talk, and I got text messages throughout it, just for the record, that said that was the best overview of UBE3A function that they've ever heard from scientists. So just so you're aware, that was amazing. And hopefully families could keep up with understanding, in summary, that UBE3A is complicated, and it has thousands of targets of what proteins it's controlling. Either it's tagging it to be broken down, or it's tagging it to be maintained. And trying to understand every downstream target is really hard. And what I learned was that there are 600 E3 ligases, and UBE3A is one of them, and this is the result of that 1 out of 600 ligases. That's only E3, not E1 and E2, which shows the power of one gene, which is pretty incredible. Therefore, we need to replace that one gene, and then we can ultimately affect all the targets. But you need to understand the complication of this, which is why when we think about how we approach and fund research, we have to think about what is the impact that that research could have on individuals living with Angelman.